The overall goal of this procedure is to transmit multiple individual spatially separated light signals through a single multimode fiber while compensating for the light distortion by mode conversion inside the fiber.
This novel technique can help to answer key questions in biomedicine and communication engineering, where multiple biological cells or data signal channels have to be addressed individually.
The main advantage of this technique, which enables spatiotemporal light modulation, is that only a single spatial light modulator is needed to both calibrate and transmit multiple independent signals.
The implications of this technique extend towards non-invasive studies and prospective therapies of neurological dysfunctions like Parkinson's disease, where monitoring and control of neurons is accomplished using spatiotemporal light signals.
Generally, individuals new to this method will struggle because of the high effort for alignment of the optical paths, which is inevitable for a high-quality transmission.
The experiment takes place on an optical bench. The optical elements have already been arranged. Some elements are only used for calibration. This spatial light modulator is among the elements used for data signal transmission on the proximal side of the setup. This CCD camera is the data signal receiver on the distal side. The data signal travels through this multimode optical fiber connecting the two sides. This schematic provides an overview of the apparatus and identifies the proximal and distal elements. A laser provides a collimated coherent light beam, which is